All right. Good morning, good afternoon, everybody. Thank you so much for joining us today. I'm going to talk to you about adaptive sampling, which is Oxford Nanopore's on-sequencer targeted enrichment technology. But I thought I would just start with a brief overview of the technology itself, because it is quite different. So in this figure on the left, you can see a sort of static image of a pore. That is a nanopore with DNA passing through it in the flow cell. And then in the middle, you can see that a flow cell essentially consists of hundreds or either thousands of these pores arrayed into a membrane on a flow cell. And so the flow cells have varying numbers of nanopores in them, and that determines the output and therefore sort of the throughput of information that you can get and the sample throughput. A bit about the sequencing itself. So we take the DNA. It is sort of tethered to a pore by this little tether, by a tether you can see on the left with a little purple head and a white line, just about here. And so this passes through the pore, and in the middle, you can see that as it passes through the pore, we also pass an electric current through the nanopore. And really, it is the effect of the DNA, each DNA base, as it changes that flow of current through the pore. That is the signal that we are reading. And you can see in the image on the right here, this sort of blue squiggle. Each squiggle represents a base as it comes through the pore, and we measure the current, and that tells us what base we are looking at. So one really exciting thing about our technology is that you don't actually have to do PCR. So you can take native DNA, you shear it to whatever length you want. So I know we typically think of Oxford Nanopore as a long-read sequencer, but we can actually sequence any read length, so from self-read DNA all the way up to ultra-long sequences. And so we don't have to do any sort of PCR amplification. So we have no bias in our system, and we're doing sort of native raw read accuracy when we're doing our alignments. And the benefit of not having to do any PCR is that we also include methylation in our sample, because we retain that information and we can read that methylation signal as a change to the squiggle. We can read both 5-MC and 5-HMC, and then with RNA, we can read all different kinds of modifications. A couple of other features that are really exciting. Sorry, my slide has not progressed. So, okay, there we go. So a couple of other things to point out. So the pores are actually live. So what that means is that the flow cell is not just a one and done use. As long as the pores retain activity, they can be reused. So you can wash them and load different libraries on, or you can wash them and load the same library on. So if you want to increase coverage or yield in some way, you can essentially just load another set of the library onto the flow cell. Or if you don't have enough samples to fill up a flow cell, you can just run the ones that you have, and then your flow cell still has capacity and sort of life, as it were, to run again. The other piece to point out is that we call this squiggle really in real time. So we do real-time base calling. So you don't need to wait until you've got to the end of a run to do your base calling and understand what data do I have. You get that data in real time, and I will show you some of the ways people are using that. So we've taken this technology and we really have sort of two core flow cell sizes. So we have the MinION flow cell, which has like 512 channels, or the PromethION flow cell, which is our larger output. And then we have different devices that can run these flow cells. So the MinION runs on either the Mark 1D, which you can see here on the left, or on the GridION, which is this one in the middle here. They are essentially the same flow cell holder, just a different throughput capacity. So we run one flow cell at a time on the Mark 1D, whereas the GridION has 5 flow cell slots. And then we have the PromethION, so that can run either on the P2 devices, so that's a PromethION with 2 flow cells, as you would imagine, or the P24, which is our sort of higher output sequencer. The other thing that I would add is another unique feature of our technology is that the technology is really held in the flow cell. The sequencer itself is a sensing platform. There are no fluidics in there. So from an installation and sort of lab space perspective, even our biggest platform is a benchtop sequencer. And just for reference, one PromethION flow cell can sequence a 30x whole human genome. So there are as we move towards more clinically focused markets, the ability to swap out a faulty flow cell, for example, with a new flow cell, rather than having to wait for an engineer to come and look at fluidics, is quite attractive. And so the other piece just to mention is that when we have these sort of multi-flow cell systems, you have complete flexibility in how you use them. So each flow cell functions completely independently. You don't have to batch. So you can load a metagenomic sample on one flow cell and a whole human genome on another flow cell and have them running completely independently. So there's lots of flexibility there. And we also have a really nice desktop application for sort of the secondary analysis, which is called Epitome. It's a really nice intuitive interface. We have pre-configured workflows, so you can see some of them listed on this screen here. And it really just for those who are not bioinformaticians or don't have teams of bioinformaticians, it gives you a really easy way to get to your answers. All right, so that's the sequencing side of things. You're here to hear about adaptive sampling, so let's dive into that. So adaptive sampling is a methodology that allows us to do targeted sequencing via enrichment on the flow cell from a whole genome library. So your library prep is very simple. It's a whole genome library prep upfront. Because we have real-time base calling, what we do is give the sequencing software a bed file, and that bed file defines the regions that we're interested either in enriching or depleting. So in the case of enrichment, we care about, let's say we have a hereditary cancer panel. As we start sequencing, so you can see in this bottom image here, as we start sequencing, we take the first 400 base pairs or so of that strand of DNA, and we align it back to the bed file. And if it matches, then we keep sequencing. And if it does not match, we reject it. And you can see in this image at the top that we literally spit it back out of the pore. So what that means is that we get a longer read, so we shear to 7 to 8 kb for adaptive sampling usually. So we have sort of these longer reads that are on target, as you can see in this darker blue color, that align to our regions of interest. But what we also get are these short sort of 400 base pair reads that represent off targets. But those off targets are actually really useful to us, and I'll talk a bit more about that. So the whole method is a very simple whole genome library prep. It's PCR free, so we have methylation retained in the information. Because the target regions are defined by a bed file, it means that if you want to run a different panel or add a new gene, you don't have to go through the whole process of getting a whole new probe set or finding a different panel and then optimizing that performance. You just add that gene to the bed file and your wet lab protocol stays exactly the same. It's really easy to update because it's just a bed file. It's not probe design. You can do it completely independently. And that whole genome backbone that I recommended provides us with the information to get a copy number. So we have a low pass. Essentially what you get at the end is enriched reads of interest and then a low pass whole genome or whole methylome. So what that looks like in practice, this is a snapshot from our hereditary cancer panel that I'm going to talk about in a moment. But in practice, what you get is this sort of low pass genome or methylome. Depending on how many samples you multiplex is going to depend on the coverage that you get. But in the case of hereditary cancer, we're in the sort of 5 to 10x for this low pass. And you can see really nicely here that this is a female. And so we have half the coverage of the X and Y chromosome as we do for our chromosomes 1 through 22. I don't know if you can see the resolution on this, but below this sort of red line, most of these are circles. That represents our sort of the off-target reads. So these are not things that we were targeting. Whereas these X's that are above the line up here, that are in the sort of 50 to 60X, these represent our on-target reads that we were targeting for enrichment. And so we have 50 to 60X coverage of the genes of interest, and then this backbone. And then you can see actually really nicely here that we have sort of 20 to 30X of our sex chromosome targets because we only have one of each chromosome. And then this just shows in a little bit more detail that we need sort of much higher depth coverage to call things like single nucleotide variants and breakpoints and get phasing. But with this sort of low, very low pass information, we can pretty clearly see copy number identification. So when we compare adaptive sampling to other targeted enrichment approaches, it adds a lot of flexibility. So for amplicons, typically we think of those as relatively small panels, so usually under 75 genes or so. They have varying read lengths, but it's mostly they are reasonably short, and it is difficult to get an amplicon panel that gives you very long reads. They are simple and easy, which is why they are popular, but they really only give you good SNV and structural variant calling. Hybrid capture gives you much more range, so typically they're much larger panels. Because you do a whole genome library prep and you sort of fragment the DNA, you can do longer reads. So it's just a pull-down approach, but it's much more cumbersome in terms of library prep. So 2 to 3 day library prep, and again, you're really only getting your SNVs and SVs robustly. With adaptive sampling, we can technically target from one base pair up to 10% of the genome. Now, in reality, you're probably not going to target a single base pair, but you could do a couple of gene panels. But I would say realistically, the sweet spot of adaptive sampling is a sort of larger gene panel because it doesn't scale linearly the way that probes do. So by adding genes, you don't decrease your sample throughput. Essentially. We can shear to any read length you want, but typically we have found that the sweet spot between getting the most enrichment and the best output of your flow cell is at 7 to 8 kb read lengths. And so that sample prep is, it's a native whole genome library prep. It's around 4 hours. And from that, we get our SNVs, our structural variants, our copy number, and our methylation. Just to hammer home the unique features of adaptive sampling. So I talked about enriching. That's how we most frequently think about it. But you can also tell the sequencer that you don't want particular regions. So if you have tried to if you have a very abundant transcript, for example, that you're not interested in, or a very abundant, sequence that you're not interested in, or you have host DNA that you've tried to deplete, but there might be some left, you can tell it that you don't want to sequence a particular region, and so it would spit those out and sequence the rest. I said that this is, very easy to make rapid edits. The off-target reads, unlike with probes where they're just wasted sequencing, here they're giving us our copy number. We retain the methylation, and because we have those longer 7 to 8 kb reads, we can get haplotype phasing, which really helps with things like pseudogene disambiguation. So I'm going to talk a bit about some of the end-to-end workflows that we've launched recently, so a couple of panels that use adaptive sampling, and then I'll show some examples of how people have made their own custom adaptive sampling panels, and really the difference is you're bringing your own bed file. For our optimized workflows, we have recommended extraction protocols to get the best read length of DNA. So one thing to keep in mind when you're trying to get longer reads is that the DNA extraction method can be quite important. But we're also aware that people have DNA in the freezer that's already extracted. And so using a custom approach allows you to be more flexible with that read length. And in both cases, we have the epitome workflows that I will highlight on the next slide. And we're doing a sort of on-sequencer targeted enrichment. We recommend running on the PromethION, either the P2I, as we call it, PromethION 2 integrated, which has integrated compute to make, because it is quite a compute heavy, as you can imagine, where every strand we're sequencing, aligning, and then spitting back out or reading. So we recommend either the P2i or the P24, so either 2 flow cells or 24 flow cells, and each of those come with compute that we know can support the application. And then for Epitome, so for the hereditary cancer panel and for our pharmacogenomics panel that I'm going to talk about now, we have end-to-end workflows built into the Epitome platform. If you make a custom panel, then we have a human variation workflow that you can use instead. Okay, so digging into some of the panels that we launched. So earlier this year, we launched a hereditary cancer panel that we are hoping can address some of the challenges that exist in hereditary cancer testing today. So some of these are that often hereditary cancer testing is sent out, and depending where you are geographically, in the US, it's not such a problem. Turnaround times are quite quick, but there are certainly other regions where turnaround time is much slower. And so the lab and the lab is waiting a significant amount of time for just to get the data back. Also, if labs are running in-house but are not very high volume, typically with the sort of short read targeted panel approach, you have to fill the flow cell, and so you can have to wait until you have enough samples to run. So you have to batch them all together. And as I've mentioned, we can debatch. And then because there are some really challenging genes in hereditary cancer testing, even if you're running a targeted panel yourself or someone else is running it, there's often additional complexity and multiple tests are required. And so we think with hereditary cancer, we have this very simple workflow. Because we can debatch, it gives flexibility in sample volumes, and we have a really comprehensive data set with just the single assay. So this is a sort of schematic of what targeted enrichment looks like. So we've put DNA extraction on day one. This may or may not obviously be incorporated together, but often the DNA has already been extracted. So the library prep for a targeted capture panel is about a day and a half or so, it's quite complicated. There's a lot of washing and hybridizing, and if you want to change the content of that panel, it can be really laborious. The sequencing often requires large batch sizes, and again, we're only getting short reads, and that in itself has some limitations on the types of variants that we can call well, and certainly the ability to get phasing, and there is no methylation. And so there may be additional testing that is required on top of this short read targeted panel. With the hereditary cancer panel, so again, we've got the DNA extraction on day one, but our library prep, as I've mentioned, is very simple. So it's 4.5 hours or so with less than 2 hours hands-on time. So if you were motivated and were doing a DNA extraction the same day, you could do all of this in one day. We then just load that onto the sequencer and sequence for 72 hours. So you just load and go, essentially. And then we have the Epitome workflow that can do all of our variant calling. And we're running 3 samples per flow cell for the hereditary cancer panel. We recommend 3 samples per flow cell. And then you get this rich multi-omic data set out. So this is the gene list. It's 258 genes, but as I mentioned, adding those extra genes doesn't mean that you have fewer samples per flow cell. We could take half of these genes off, and we would still want 3 samples per flow cell to get to that, at least 30x coverage of our enriched regions. I have highlighted here some of the genes that are difficult and that often require additional testing, and we'll look at some data from a few of these from beta testing and from our own internal validations. So starting with the SNVs and indels, so we took publicly available Coriell samples and just confirmed that we can call all of the SNVs and then the indels that were found in those. So you can see these listed here. And as an example, I'm going to show you this top insertion, this 8-base pair insertion in VHL. So VHL is mutated in von Hippel-Lindau disease, a hereditary cancer syndrome, and you can see very clearly here in this Coriell sample that we have, you can very clearly see this 8-base pair insertion in VHL. But the really nice thing is that you can layer on the methylation. So now we've sort of collapsed down this insertion. So that's in this box here. So this is where the insertion lives. But when you layer on methylation, you can see that we also can see hypermethylation of the VHL promoter and exon 1 in this same sample. So just giving that extra richness of information for this sample. Because we have longer reads, we can capture large structural variants quite well. This is an example of a Seracare sample that has a 23 kb deletion in BRCA2. There is also another deletion in BRCA2 in this same sample, and we detect that as well. But the 23 kb one is the biggest one, so I showed that. And then I mentioned pseudogenes. So one of the challenges we have internally, obviously, is we don't have a lot of samples with PMS2 mutations in them. But one of our beta testers who has really done quite a lot of work on this panel now subsequently since it's launched, was able to get just from her beta testing some really nice data showing deletion of exon 14 in PMS2. And the important thing here is that she got haplotype phasing. So you can see that here at the top, she was clearly able to see this deletion of exon 14 in one haplotype, whereas the haplotype below is very clearly wild type. And significantly, she can see that the PMS2CL pseudogene is also wild type, so no variants detected there. And that often is the challenge with short read, is that you know there's a mutation there, you don't know which haplotype, and you don't know if it's in PMS2 or PMS2CL. Moving on to pharmacogenomics. So I'm sure most of us know that everybody metabolizes drugs differently. And 99% of individuals have a variant that means that affects how they metabolize a drug. That can be inconvenient, but the worst case scenario obviously is adverse drug reactions, which are events that we obviously want to be avoiding. And over 310 drugs have received pharmacogenomics information in their label, indicating that people will behave differently when given this drug. The challenge with pharmacogenomics genes, biology is taunting us, is that these are really challenging regions to detect variants in. They often have duplications in the presence of a deletion. They are highly homologous, they are repetitive, so short read sequencing really struggles, and testing in general obviously requires testing in general often requires multiple different tests for each allele that you will be looking at. And so with adaptive sampling, we have a pharmacogenomics panel that has over 150 genes, so it covers almost 10% of the human genome. We have the same sort of 7 to 8 kb read length. In this case, we are multiplexing 4 samples per PromethION flow cell. And the reason we are doing that is because to accurately call CYP2D6 with our CYP2D6 caller, we need slightly higher coverage than the usual 30x. And so we're actually washing and reloading samples to get that higher coverage, which means that we can squeeze 4 flow cells on. So it's kind of a balance. So we have 4 samples per PromethION flow cell, and then same half-day library prep, and then 72 hours of sequencing with loading your library as it's going, essentially. And this allows us to capture some of those really difficult variants. This slide just talks about CYP2D6, which is certainly the best known, which has 4 different challenges in detecting CYP2D6 alleles. So firstly, there is high pseudogene homology. It is really hard to phase variants with short reads, whereas longer reads make that much easier to do. We are also better able to resolve these sort of complex gene-pseudogene hybrids, and we detect copy number gains and losses from that sort of low-pass backbone that we talked about. And so this figure just shows that either with 5 kb reads, we're somewhere in the middle, but with 5 kb reads, we can span many of the genes, the pharmacogenomics genes, and with 10,000 plus, we span beyond those genes. And both of these give us really nice ability to do haplotype phasing and get copy number information and any complex rearrangements that are there. All right, so now, so those are the panels that we have developed just to sort of make it easy to get started. And we've done a lot of work to make sure performance is good. But one of the benefits, obviously, is that what changes a panel is just a bed file. So I'm just going to give you some examples of things people have done with adaptive sampling in the wild, I guess. And so just a reminder, so this is going to be the same simple 4-hour library prep and then targeted enrichment and sequencing. And we've made recommendations for our off-the-shelf panels of sequencing for really the maximum lifetime of the flow cell to make sure we get the best enrichment and the best throughput. But you'll see that people have used very varying levels of sequencing because it gives them the answer that they want. In this context, for a custom panel, you would be using the human variation workflow, or of course, if you have your own pipelines and bioinformatics, you can also use those. So the first case I'm going to talk about is a publication from Tom Walsh and Mary Claire King's group in the University of Washington, where they looked at what they call Cancer X families. So these are families that have, according to the family tree, they have a very clear germline predisposition. So there's something familial happening, but they had been tested either by whole genome, whole exome, or a targeted panel, so some combination of those things, without finding any pathogenic variants. So what this group did was they made a 10-gene panel, you can see it listed here, for adaptive sampling, and then they validated those findings with a sort of targeted long-read cDNA sequencing approach. And what they were able to find in those families is that many of them had what they call deep intronic variants. So they had to do functional studies to show that these variants that they found were actually causative. But they found that they were essentially able to resolve from 120 unsolved families, they were able to resolve like 92 causative variants that they thought were predisposing, and in some cases actually functionally determined this in 88 of those families. So really increasing their diagnostic yield from this testing. Another example is also from the University of Washington. They are looking at retinoblastoma. So one of the interesting things about retinoblastoma is that different patients can have really drastically different outcomes. And what drives that has not been well understood. So these researchers were able to utilize a differentially methylated region in intron 2 of RB1, which is the gene that is mutated in retinoblastoma. And you can see that here at the bottom. So we have hypermethylated in blue and hypermethylated in red. And you can see, and what they were able to do was use that to understand sort of parent of origin of each of these alleles without having to have a sort of, the parent sample, so no need for trio testing. And when they looked at survival rates with those, they were able to see that inheriting on the paternal side, inheriting the mutation from the paternal side is really strongly associated with poor outcomes. Whereas when you associate a mutated RB1 from the maternal side, you can see that there is a drastic difference here between maternal and paternal. So really interesting data. And interestingly, they were telling me that the RB1 is sort of close enough to BRCA that you can actually use this imprinted methylation to look at like maternal parent of origin for BRCA as well, for BRCA2 as well. This is an example from Jeremy Wang's lab at UNC. So he and Julie Guy published this really nice paper of rapid classification of acute leukemia using adaptive sampling. So they've done the DNA extraction and then the library preparation. So for them, that takes them about 3 hours. And then they are doing their sequencing. And within 15 minutes, they can essentially get enough copy number resolution to do what they call digital karyotyping. And then they continue sequencing for sort of 3 to 6 hours to get structural variants. This is just an example of what that digital karyotype looks like. So they're using it to detect aneuploidies. It looks really great. And then these are some examples of some of the large, complex and structural rearrangements that they've found. And I'm sure many of you know that DUX4 can be incredibly challenging. IGH mutations are impossible, very hard to detect, certainly by RNA, and take a lot of sequencing to detect by DNA sometimes. And then they were also able to find these ERG structural rearrangements. I'm going to switch gears a little bit and just introduce this concept of tumor methylation profiling. So typically, when understanding what type of cancer you have, there's often some, immunohistochemistry and then some molecular testing. And in the case of brain tumors or central nervous system tumors, these tests are often not in alignment. So brain tumors are particularly difficult to diagnose because sometimes they look morphologically the same and then behave very differently, and sometimes they look very different but they behave the same, and sometimes you have your IHC that tells you this is one subtype but then the molecular data doesn't agree with that. So I would say in the past 5 years it has emerged that these brain tumors all have very distinct tumor methylation signatures. This was first identified using array data, but because we offer methylation from native samples, we can get that methylation information as well. And the classifiers that were trained on this sort of EPIC array data, we get the same sort of classifications with Oxford Nanopore data. Excuse me. And so these tumor methylation signatures are emerging as a really accurate but also rapid way of getting a tumor classification from these very difficult tumors. So we will be launching early next year a fully supported protocol to help guide on like how to make your library to get that methylation classification, and then keep sequencing and get your adaptive sampling information. So you can get not only methylation, but all of your SNVs, indels, copy numbers, and structural variants as well. The University of Nottingham in the UK, who have started to use this approach in an intraoperative brain tumor setting. So essentially what they are doing is taking sample during a brain surgery, extracting the DNA. So what you can see in this figure is on the bottom, this is the path that they're following. So they take the sample from the surgery, they extract the DNA in 30 minutes, they make their library prep, and they get that onto the sequencer within about an hour and a half to 2 hours. And then they start sequencing, and typically within 2 hours, they have enough information to get a methylation classification. And then they keep sequencing overnight to get that the additional information. At the top here, you can see this was the timeline for their standard of care testing, where they had to send out these samples. And it could take as long as a month to get all of that information back. And they're able to essentially reduce that down to same day to get the methylation classification, and that can be used to guide surgery. So if it turns out that it is a benign tumor, obviously the surgeons are going to be less aggressive. If it is a very aggressive tumor, then the surgeons, that will guide how they need to behave. So really powerful. And this is just some of the data from that paper that showed that they get the MGMT promoter methylation status, they get copy numbers, they can call gene fusions, and they're calling single nucleotide variants. And then the last example that I'm going to show is a publication that came out in medRxiv in October and then was presented at a really nice poster in ASH just this past weekend, where they also used adaptive sampling to look at comprehensive pediatric cancer profiling. And what I really like was that this figure that they made is a really beautiful illustration of the sort of timeline of information that they can get. So they created this pipeline that runs all their this wrapper that runs all of their pipelines. And essentially within the first hour, they have enough information to generally get a methylation classification and their sort of large copy number variants. And then within, the next hour to 12 hours, depending on, how prevalent that is and what your tumor fraction you're looking at, they have their small copy number. They run from sort of 3 to 24 hours to get their fusions and their clonal single nucleotide variants. And then they keep running for that last, couple of days just to get the highest depth coverage they can for subclonal SNVs and indels. And so they have some really nice stories. I'm sure you don't need me to read these figures out, but where they were able to detect, multiple complex rearrangements with this single sample. So I'll just use this top one as an example, where they were able to detect a fusion, mutations in Notch1, PIK3R1, and DNM2, and then some deletions, so trisomy of chromosome 8, and then loss, gain of 10p and 19q, and then copy neutral LOH on 9p. So really a huge amount of powerful information from one simple experiment. They did a really nice analysis. This is very small, so I'm going to blow it up in the next slides. But essentially at the top, this shows what their sort of standard of care testing was. And then they compared to, did they call fusions, SNVs and indels, and then their copy numbers. So just digging into that a bit more so those of us who need glasses can actually see what's happening. So at the top, this shows the testing that they've done. So they had various different pediatric cancers, mostly heme, but a couple of solid tumors as well. So they had BALL, TLL, Burkitt's lymphoma, AML, MDS, and then some solid tumors. The gray boxes in this top section here mean that this testing was done on those samples. So most of these got exomes, except for this one here, for example. So just to orient you, that's what you're looking at. And then this first figure just shows, were they able to detect the gene fusions where they had found them? And, the answer was generally yes. There was one example here in yellow where they only had 1 to 7 reads, and they set 8 as their cutoff for a confident call. But they did see the fusion in their data. This is their SNV and indel information. So again, same idea. Blue means that they called it. There was this one sample here that I believe he said was mosaic. It was a very busy conference. But you can obviously refer to the publication to check. And then looking at copy number, so we have sort of, they've divided it into sort of their large copy number, so hyperdiploidy, CNV, and LOH, and we're able to detect everything but one CNV event in this MDS sample, and then one small CNV in a BCL. And this one was a sort of, RB1 was a sort of borderline case. But as a first pass of their data, it was incredibly powerful. It was really exciting to see. So that is everything that I have.
Thank you to Claire for the presentation, and thank you all for joining us. If you'd like more information about today's broadcast, please use the QR code on your screen or email support@nanoporetech.com. Thank you all once again, and enjoy the rest of your day.